We've all been affected by neurodegeneration in some form or another, whether it's ALS, Alzheimer's, Parkinson's, autism, or MS. We've known family members, friends, or maybe we ourselves have been affected by these disorders in some way or another. I'm Jim Monser, founder and CEO of RLS Therapeutics. In my 42-year career in life sciences, biotechnology, pharmaceuticals, medical devices, I never thought that I would be a candidate to use some of the products that I was developing. But in 2017, I was diagnosed with Parkinson's disease. That was the trigger point for me to reach back out to Bob Hoffman. Dr. Bob Hoffman is the inventor of this compound. I met him back in 2002 when he was working on developing this compound. He initially designed it for treatment of dry form macular degeneration. He was sick and tired of treating his patients with just steroids, which are only symptomatic treatments and don't cure the disease. So he developed this compound and, under physician consent, asked his patients if they would be willing to be treated with this compound. 26 patients signed up for it, and on average they could read 5 lines further down the chart. But more significantly, the drusen, which is the inflammation in the retinal cells, was gone. So Bob was pretty thrilled with those results, and he was talking with a friend of his who at the time owned the largest equine hospital in East Texas, a fellow by the name of Dr. Terrell Buchanan. So Bob and Terrell were talking about this compound that he had been developing, and Terrell suggested they try it with horses who have an equine disorder that's much like Parkinson's or ALS. It's called equine protozoal myeloencephalitis, or EPM for short. So Bob and he designed a study for treating 311 horses, some of which were so impaired they could not walk into the barn by themselves. They needed people to hold them up just to get them into the stall. The results were pretty stunning. 3 weeks out, 95% of the horses had no symptoms at all. In fact, horses that could barely walk in the stall were now trotting around the corral and running on their own. It was pretty astounding. So I met Bob in 2017 after I got diagnosed with Parkinson's, and we reconnected and began developing this compound for human indications, but also for these animal health indications like EPM or small animal neurological disorders. So since then, we've raised $1.7 million, and we've done several things with that. We repeated the horse studies that Bob did 25 years ago with the same astounding results. This time they were under the care of Dr. Steve Reed, who is one of the top equine neurologists in the world up at Root and Riddle Animal Hospital in Lexington, Kentucky. We've run some cell culture studies in the lab that have astounding results. The net of all of those studies is that we have confirmed that it is highly likely that our compound will have very compelling efficacy against the major causes of neurodegeneration. So let me go back through those studies in more detail. So under Steve Reed's direction, we've treated 20 horses, 14 of whom have confirmed EPM diagnoses. All 20 horses had very significant improvement. Again, the same kind of outcomes. Horses that couldn't walk, or some of which were hooked up to be euthanized that very day, now about 6 weeks later are walking around in the corral by themselves, and a couple of them actually can be ridden, and one I think has returned to competition. The cell culture studies. The first one utilizes what's called the luminescence bioassay. The bottom line is that measures the mitochondrial health of the cell, or the metabolic health of the cell, which is critical to the function of every cell in your body. With this study, we treated these human cell lines, both they were cardiac, liver, kidney, and brain cells, with toxins that are available in the environment, several toxins, one of which is called rotenone. Rotenone has been linked to Parkinson's. And what we showed in our study was that we could recover the function of brain cells that have been damaged by rotenone, kind of reflecting the disease course for Parkinson's or ALS. We followed that with treatment of our compound and showed not only recovery to untreated baseline, but to 10 or 15% above untreated baseline, which is pretty astounding. It shows that we can recover mitochondrial function in cells that have been damaged by this toxin. The next study was what's called a seahorse assay. And the name isn't important, but what that does is it measures the metabolic function deeper into the mitochondria at the electron transport chain level. And that's the core biological process that converts all food into energy in your body, and therefore it drives the ability of the cell to do what it's supposed to do. So we designed that study the same way we designed the cell culture study, the luminescence bioassay. We treated with rotenone, showed the decline in function, came back and treated with our compound and showed recovery to above baseline. The compelling part of all of this is that all neurodegenerative disorders have these common factors in terms of root causes. And they don't know necessarily which one triggers first, but they know that they all work in cascade to take these cells down to a dysfunctional state. They are neuroinflammation, mitochondrial dysfunction, and protein aggregation. And our studies show that we check the first two boxes in a very compelling way. We are very excited about these results. They show that our compound could have very compelling efficacy in treating these major neurodegenerative disorders. Our business model is to develop technologies like this out to the point where it's a compelling data package. Typically for a pharmaceutical product, it would be somewhere in the clinical trial phases. And then we sell that or license that to a big pharma who has the wherewithal to execute the large-scale clinical trials, to do the commercial launch, med ed, reimbursement, marketplace presence, the whole thing. So we would license these compounds and the rights for these diseases to a big pharma who can go execute. In the same manner we do that with animal health as well. So we're raising $4 million, and the purpose of the $4 million raise is to get us to two outcomes. One is, we've met previously with big pharma companies like Johnson & Johnson Innovation and also some animal health companies, and they told us basically the same thing. We need to have this core package of data, a new batch of the old material, some formulation data, some basic safety and efficacy data, and some mechanism of action data. So we have a plan to run these studies that will get us all that package of information, potentially as fast as a year and a half to 2 years, which gets us back to the conversations with these companies about potential research collaboration or licensing agreements. Short of that, there's a shorter list of studies that may allow us to get to what's called compassionate use treatments. And this is under the FDA regulations where a physician can treat patients who are in advanced stages of these neurodegenerative disorders, who have no recourse. They're going to die soon. They've tried all the available therapies. They can't get into clinical trials, and they are desperate for something to try. And under compassionate use or expanded access, a doctor can take unapproved drugs and treat these patients under FDA approval. So to get to that list of studies is about a $2 million raise. We're obviously going after the $4 million raise, but the $2 million raise is a starting point. If you're interested in this investment opportunity, we would love to hear from you. We feel that this is a remarkable opportunity to have a major impact in human health, but also deliver remarkable returns to investors. If you're interested, please reach out to me.